What controls cell division? Well, one of the things is that cells divide at different rates. So certain cells are going to divide way faster than others. For example, skin cells divide very quickly. And then cell size controls cell division. If the cell isn't big enough, it won't divide. But if it gets too big, it's going to die. So the size of the cell matters. So here's some examples of different cells and how quickly they divide. Here are some plant cells. It's easier to see plant cell division because of the cell wall. Notice like prophase, the chromosomes are condensing. It's very different than interphase, where you can see the nucleus of the cell, the nucleoli, and the nucleus of the cell. Plants have multiple nucleolus, so don't get confused by that. Metaphase, they all line up in the middle. Anaphase, they start to move away. And then telophase, you would get 2 new cells. The haploid number is half of a diploid number. So when we're talking about dividing up chromosomes, diploid number means we've got duplicated chromosomes. But a haploid number, we only get half. So meiosis is going to take a sex cell and divide that sex cell so there's only half the total number of chromosomes. So sperm only has 23, whereas skin would have 46. So in this case, if it's a haploid number, you want to multiply by 2. If it's a diploid number, you want to divide by 2. So you pause the video and complete that chart. This is a table that just compares mitosis and meiosis. With mitosis, there are 4 different stages, P, MAT. With meiosis, they go through those stages a second time. In mitosis, it's somatic cells, body cells. With meiosis, it's sex cells, gametes. With mitosis, the final result is 2 new cells. They are identical. With meiosis, there are 4 cells. They are not the same. In mitosis, we get diploid cells. In meiosis, we end up with haploid cells, half the total number of chromosomes. In mitosis, there's no crossing over. The chromosomes do not swap genetic material. In meiosis, they do swap genetic material. In mitosis, they're just clones. There is no variation. But with meiosis, there is some variety. The daughter cells are not like the parent cell. Here's what we mean when we say homologous chromosomes. Same size, same shape, same genetic material. You'll notice that there's like a left side to the chromosome and a right side. So these histochromatid just means that's the normal chromosome and it's now been replicated or copied. During meiosis, those homologous chromosomes will pair up, they line up. And this is when sometimes there's some crossing over. All right, we can swap some of that genetic material. That's what that looks like. So homologous chromosomes will sometimes swap genetic material. And as a result, there's an increase in genetic variation. This only happens during meiosis and only when they're lined up. When we look at something like a plant, we might think, well, we don't know anything about this plant. How do we know what divides by meiosis? Well, that's fine. We don't need to know anything about the plant. All we need to know is that meiosis has to do with gametes. Gametes are sperm and egg. So we look at the parts of the plant, we see something like ovary contains eggs. Well, those are going to divide by meiosis. Or when we look at the anther makes pollen-containing sperm, sperm is going to divide by meiosis. So without knowing much about plants or flowers, we can still answer a question about meiosis. Here's the steps of meiosis. It's going to be PMAT, but twice. All right, so here's a prophase where we can see the chromosomes, they're duplicated. Here's a metaphase, they're lined up in the middle. Anaphase, they start to move away to either side. A telophase, we've got 2 new cells. But meiosis will go through the process again. We'll have another prophase. We'll have another metaphase. And then down here we have anaphase. Notice that with anaphase, we have a smaller number of chromosomes. All right, so we just have the one chromatid, not the whole chromosome. They've split. This is how we get the haploid number. So we've got telophase part 2 here, in this case, 4 new cells. A karyotype is a way we can just look at the chromosomes in those new cells. Notice that we have an extra chromosome for number 21. What that means is during meiosis, some of those chromatids did not separate. They stayed together. And as a result, when that egg was fertilized, it got an extra chromosome. Number 21 means it's Down syndrome. The chances of this happening, this process is called non-disjunction, the chances of it happening goes up with the mother's age. The older the mother, the more likely there's going to be a problem with non-disjunction. So hopefully that kind of helps you understand a little bit about mitosis, meiosis, and the case that you're reading about.